Here's a question that might sound ridiculous. What if I told you that the same packet of yeast sitting in your kitchen drawer right now, the one you use to make bread, could wake up dying plants, could push roots deeper into soil than most commercial fertilizers? You'd probably think I was exaggerating, but here's what's strange. Scientists have actually tested this. Peer-reviewed journals have published the results, and the numbers are uncomfortable for anyone who's spent serious money on garden center products. In controlled studies, crops treated with simple yeast showed yield increases of 20 to 30%, root biomass jumping by double digits, plants pulling in nitrogen and phosphorus at rates that surprised the researchers running the experiments. So why isn't this on shelves everywhere? That's the part nobody wants to explain. Let's start with what baker's yeast actually is, because most people have no idea. Saccharomyces cerevisiae. That's the scientific name. It's a single-celled fungus that humans have been using for over 5,000 years. Archaeological evidence confirms we were baking and brewing with this organism before we had written language to describe it. But here's where it gets interesting for gardeners. When you crack open a packet of dry yeast and add warm water and a bit of sugar, you're not just activating it, you're triggering a metabolic explosion. Those dormant cells wake up hungry, they consume the sugar, and as they do, they release a flood of compounds into the water around them. B vitamins, specifically thiamine, riboflavin, niacin, pyridoxine, and folic acid. Saccharomyces cerevisiae synthesizes these internally, and scientific literature confirms it's one of the richest natural sources of B-complex vitamins available. Amino acids. The protein content of yeast is roughly 50% by dry weight, and when yeast cells break down, they release free amino acids that plants can absorb directly. And here's the part that matters most: plant hormones. Studies have documented that yeast metabolites include compounds like auxin and cytokinin, the same hormones that regulate cell division and root elongation in plants. When roots encounter these signals in soil, they interpret them as instructions to grow. This isn't alternative medicine, this is biochemistry. In 2014, researchers at a university in Australia published something fascinating in the journal Crop Science. They took brewer's yeast, the same Saccharomyces cerevisiae found in baker's yeast, and applied it to sugarcane and tomato plants, both living yeast and dead yeast. They wanted to know what would happen. The results were striking. Plants treated with yeast showed significantly higher root biomass than untreated controls. The nitrogen and phosphorus content in both roots and shoots increased measurably. And here's what made scientists pay attention. They observed yeast cells actually being taken up inside plant root cells. The researchers documented a phenomenon called rhizophagy. Plants were essentially eating the yeast and extracting nutrients from it. A separate review published in 2025 in the Journal of Pure and Applied Microbiology compiled data from multiple studies. They found that yeast applications resulted in yield increases ranging from 20 to 30% in crops like wheat and sugar beet. In fava bean trials, plants inoculated with yeast species showed dry weight increases of 8 to 17 grams per plant compared to controls. A 2024 study on lettuce found that a 3% yeast solution produced the highest chlorophyll content, the longest plant growth, and the heaviest root and shoot weights compared to other concentrations. These aren't fringe experiments. They're published in agricultural journals, peer-reviewed, repeatable. So here's the uncomfortable question: if the science exists, where are the products? Walk into any garden center and you'll find shelves stacked with fertilizers, bottles, bags, pellets, granules, products engineered to sit in warehouses for months or years without changing. That's not an accident. It's a business requirement. Chemical fertilizers are built for stability. The salts inside them don't degrade. They don't spoil. A bag manufactured 2 years ago performs identically to one made last week. That predictability is what makes the entire supply chain possible. Now think about what happens when yeast meets sugar and water. Within minutes, fermentation begins. Carbon dioxide gas starts building. The mixture becomes biologically active, which is exactly what makes it valuable. But that activity comes with a cost. If you sealed that mixture in a bottle, the pressure would build. Home brewers know this danger intimately. Glass bottles can shatter. Plastic bottles bulge and deform. People have been seriously injured by what brewers call bottle bombs, containers that explode when CO2 pressure exceeds what the material can handle. And even if the bottle survived, the mixture would spoil. Within days, bacterial contamination takes hold. The smell shifts from bready to sour. The beneficial compounds degrade. There is no preservative strong enough to stop active fermentation without killing the yeast entirely. And dead yeast in a stable solution loses most of the signaling compounds that make live yeast valuable to plants. This isn't a conspiracy, it's logistics. You cannot mass produce, ship, warehouse, and sell something that's actively alive and fermenting. The business model doesn't work. The liability doesn't work. The shelf life doesn't exist. The only way to use this method is to make it fresh, use it immediately, and never try to store it. Which means the fertilizer industry has no product to sell you. Here's the method that research supports. Start with 1 liter of lukewarm water. Temperature matters here. Too hot, around 120 degrees Fahrenheit or above, kills yeast cells. Too cold, below 70 degrees Fahrenheit, keeps them dormant. You want water that feels comfortable to the touch. Body temperature is the benchmark. Add 2 tablespoons of active dry baker's yeast. That's roughly 20 grams. This is your population of living cells. Add 1 teaspoon of plain white sugar. The sugar is fuel. It has no direct benefit to plants. It exists only to wake up the yeast and give them something to consume during the fermentation window. Stir until dissolved, then cover the container loosely. Not sealed, never sealed, just protected from dust and direct light. Wait 30 to 40 minutes. You're looking for foam. Bubbles rising to the surface, forming a frothy layer on top. That foam is carbon dioxide being released as the yeast metabolizes sugar. It's visual confirmation that the brew is alive and active. If there's no foam after 40 minutes, something went wrong. The yeast was too old, the water was too hot, or the sugar was missing. Once you have active foam, dilute immediately. Take that 1 liter of concentrated yeast mixture and add it to 10 liters of plain water. This 10-to-1 ratio isn't arbitrary. Undiluted yeast water is too strong. It can overwhelm delicate root hairs and actually stall growth instead of promoting it. Use the diluted mixture right away. Water directly into the root zone, not on leaves. And discard any leftovers. This isn't something you can save for next week. Here's where I need to be honest with you. This isn't magic. It won't resurrect dead plants. It won't turn clay soil into garden paradise overnight. And it definitely won't replace proper watering, sunlight, and basic plant care. The research shows the most dramatic results in specific conditions. Nutrient-poor soils respond best. Sandy soils that struggle to hold on to minerals. Depleted beds that have been overworked. In these environments, the vitamins, enzymes, and hormones in yeast water can act as a jumpstart for root development. Fast-growing plants tend to react more visibly. Tomatoes, beans, lettuce, peppers. Species that already want to grow quickly seem to pick up the signal and run with it. But in rich, fertile soil that's already teeming with organic matter and microbial life, the effect may be subtle. You're adding to a system that's already functioning well. Soil that's too wet creates problems. Yeast can overgrow in waterlogged conditions, and mold becomes a risk. Heavy clay that stays compacted may not benefit much regardless of what you add. The visible signs that it's working? Fine white feeder roots appearing near the soil's surface within days, leaves taking on a deeper green, plants standing taller, more upright. Sometimes a faint sweet smell near the base immediately after application. If you see roots browning at the tips or leaves wilting after application, the solution was probably too concentrated. Dilute more next time. Not every garden will respond the same way. Not every plant will show dramatic changes. The best results show up where the soil is hungry and the plant is primed for growth. Here's what I find fascinating about all of this. We've spent decades engineering increasingly complex fertilizer products, slow-release pellets, targeted nutrient blends, synthetic formulations designed in laboratories, and the entire time, one of the most extensively studied organisms in biology, an organism we've worked with for 5,000 years, has been sitting in kitchen drawers everywhere. Saccharomyces cerevisiae was the first eukaryotic organism to have its entire genome sequenced. Scientists have used it to understand everything from cell division to human aging. We know more about this single-celled fungus than almost any other life form on Earth, and yet most gardeners have never heard that it can improve root growth. The research exists, the mechanism is understood, the method costs almost nothing. What it can't do is sit on a shelf. It can't be bottled, branded, and sold at a markup. It can't generate recurring revenue for anyone, so it stays invisible. But now you know. A packet of yeast, a teaspoon of sugar, some warm water, and 40 minutes of patience. That's the recipe the garden center doesn't sell. Try it, watch your roots, and see what happens underground.